So without further ado, first up we have Junyuan Cao from the University of Tongyi in Shanghai, who will be speaking to us today about fecal pellet flux in the Western China Sea. So take it away.
Here's my share screen. Is that okay now? I'm sharing, right?
Yep, we can see it.
Okay, so I can get started, right?
Yep, whenever you're ready.
Okay. So, hello everyone. It's really nice to meet you all, and it's my great honor to be invited to share my work on Jack Zone seminar. So my name is Junyuan from China, and I'm a PhD candidate student in my fourth year of my study. So today I am here on behalf of my group to introduce our recent work about the fecal pellets in the Western South China Sea. And this work is based on our recent published paper entitled Temporal and Vertical Variations in Carbon Flux and Export of Zooplankton Fecal Pellets in the Western South China Sea. And first, I would like to introduce why we conduct this work. So as we all are very familiar, the biological carbon pump is a process that atmospheric CO2 is observed by phytoplankton, and they are exported from ocean surface to the deep sea in different forms and through different mechanisms. So in the downward process, the particle organic matter plays an important role, and we know that TOC include phytoplankton like detritus and zooplankton carcasses and organic aggregates. They all play important roles. And also as well, today I would like to focus on zooplankton fecal pellets. As one component of particularly organic matter, fecal pellets are able to carry the POC in the thinking process from surface to the deep sea very with high efficiency because we know it has a fast thinking velocity. So since 1970s, the fecal pellets have been extensively studied, including their, for example, their shape, their color, their size, and their inner contents. Also, about their production, sedimentation, and fate during the sinking process, as well as their vertical sinking flux. So, among these studies, I think zooplankton fecal pellet fluxes have been widely investigated, which we can see it exhibits great spatial and temporal patterns in different regions all around the world. However, I think there are two limitations. We can see in these compilations of previous work, we can see the studies paid more attention to the middle and high latitude areas, and they focus more on the shallow water depths about the production and retention of fecal pellets in the surface ocean. That's why we try to extend the work to a new zone, to the tropical ocean and into the deep ocean. So I think in the second part, I would like to introduce my study areas, the South China Sea. South China Sea is the largest marginal sea, which it separates the Asia land from Pacific Ocean. And one important background I would like you to know is that the East Asia monsoon system is the prevailing climatic controlling factors in the South China Sea. And the strong winter monsoon typically lasts from November to March or April, resulting in the northeasterly winds across the whole South China Sea. We can see, I think, the blue arrows. And while the summer monsoon lasts from June to September, it resulting in the southwesterly winds across the South China Sea. And another background is about the From other researcher studies, the zooplankton abundance in the whole South China Sea and in our study areas dominated by copepods. So my work focused on the western part here of the South China Sea, which is located between the northwestern continental shelf and the deep basin of the North-South China Sea. And here is the zoom out picture. You can see there are two mooring systems we called TJW1 and TJW2. They were deployed at the west of the Sanquan submarine platform in the western South China Sea. And you can see there are 2 sediment traps tied at 500 meters and 1,170 meter water depth on the TJWE mooring, and there was only 1 sediment trap 1,380 meter water depth on the TJW2 mooring. So totally these 3 sediment traps, we can see it forms a vertical transect with the increase of water depth. And for each sediment traps, we collect 21 time series samples from May 2021 to May 2022 with a 17-day intervals. And so totally we have 21, we have 63 samples in our study. And in this slide, I would like to briefly introduce how we conduct our experiments in our lab. So first, the sample was split into a series of alicos after 1 millimeters wedge sieving. And 1 alicot for each sample was preserved for our zooplankton fecal pellet studies. And the preserved samples was then saved through a 20 micrometers mesh. And after that filtration, we resend the samples onto this graded Petri dish, and we depose the dish, and we observe under this stereotype microscope equipped with a digital camera, which make us to take photos of each grid. So each grid was photographed at small magnification to count all the large fecal pellets. And then in each grid, the pre-measured pairs were randomly chosen and photographed at a larger magnification to count enough small fecal pellets. So all the fecal pellets can be categorized into 3 shapes, which are ellipsoidal, cylindrical, and spherical in our study areas. Then we separately counted different shapes of pellets in all the photos. So considering the areas of the whole Petri dish and the squares we choose, the fecal pellet number can be extrapolated to the whole sample. Then the geometric parameters of randomly chosen fecal pellets were measured from all photographs. We use the Fuji software, might also be known as ImageJ software. And the biovolume was calculated from the formulas of the different shapes. And we get the biovolume of each shape of fecal pellets. Then the biovolume was converted to carbon content to obtain the fecal pellet carbon flux using this conversion factors and 0.036, and this factor was tested by one of my colleagues 2 years ago. Yeah, so in my study we just use her factor. Then I think we can move to the result part. And as I have said before, there are totally 3 types of zooplankton fecal pellets including the spherical ones, the cylindrical ones, and the ellipsoidal ones. Yeah. So they are, I think the color is from light brown from light green to green brown and to dark brown, I think. And we listed all the parameters of pellets in our photos pictures. And I won't read it through, but I want to point out two trends of biovolume. First is that it's very obvious that the cylindrical pellets have the largest biovolume, and then ellipsoidal ones have the large range. For example, you can see this one is pretty large, but also have the smaller ones, and maybe smaller than this one I pointed. I mean, the ellipsoidal pellets has a very large range of biovolumes, and the spherical pellets are generally small. And this one is relatively large ones. Yeah, they are pretty small. And also the biovolumes of all kinds of pellets, they increase with the water depth. And the second part, I would like to introduce the fecal pellet numerical flux. One of the very clear result is that fecal pellet numerical flux decrease with water depth. You can see in 500 meters, it was averagely 7.9 thousand individuals per day per square meters, and it decreased to almost half to the deepest trend. And if we see the result time seriously, at 500 meters, the fecal pellets was little and was little in the summer to autumn, except for one peak value in the late July. Yeah, and once entering the winter monsoon seasons, you can see there first it occurred a very surge increase at around November, early November, and then during the whole winter season, it was remained pretty high. And after the winter season monsoons, it decreased a little. And at 11.7 hydrometers, the highest FPN flux occurred in August, and it fluctuated in the remaining of the year. And you can see a difference is that FPN flux at this water depth, it showed very low values in winter season. And at 13.8 water depth, it also has small fluctuations all year round, and it's also low in the winter. Another phenomenon is very interesting that in the two deep water depths in the last samples in early May, there is a surge increase. We don't know why, but it's a very interesting phenomenon. And if we look into the different types of fecal pellets that contribute, how the different types of fecal pellets contribute to the overall FPN flux, the result is that the ellipsoidal ones was the most abundant. You can say it constitute of 50 to 60%. Then the spherical was also pretty abundant to about 30 to 40%, and the cylindrical pellets was very scarce, only 3 to 5%. Oh, yeah. Then we move to the fecal pellet cover flux. We can see the FPC flux were very similar at 3 water depths. The average value was very similar. And if we see the time series result, it's pretty like the FPN result as I talked before. At 500 meters, it was averagely high in winter, but also has the two peaks in late July and was entering the winter monsoon around November. At the second water depth, it was highest in the August and also had a peak was entering the winter monsoon seasons and then remained low in the whole winter season. And the deepest centimeter trend, I think the result shows it fluctuated all year round, with no clear trends of which season is more and which season is less. And if we see what different types of fecal pellets contribute to the overall FPC flux, there is an interesting result. If cylindrical pellets increase a little. If you remember, the cylindrical pellets just accounted for 3 to 5 in the number, but it accounted for like 20% to 30% in the FPC flux. That means for each individual pellet, the cylindrical ones are large. Yeah. So we have the above are the result parts. So we have all these data sets, and we made 3 discussions based on the data I introduced. So the first one is we want to see the contribution of fecal pellet carbon flux to the overall POC flux. I didn't introduce POC flux before, but I plotted in this picture with light green bars. So the result here is the overall FPC over POC flux ranged from 0.7 to 28.2%, yeah, with an average value of 4.4. And I think it has no clear time series patterns when we look at the 3 water depths. But one interesting result, I think it more or less matches the negative correlations between POC flux and this FPC to POC ratios. And this theory is already raised by Wilson in about 10 years ago. And for one extreme example, in the at 500 meters in the January, we can see the POC flux was very, very high, but the FPC flux was just normal. It didn't increase a lot. So in that month, the FPC over POC ratio, it reached the lowest point that 0.68. Yeah. So that means sometimes when the ocean primary reflection is high, it didn't contribute by fecal pellet, maybe some other components. And if we compare our data 4.4 on average to the worldwide data, I think we can see that our data laid in the reasonable but relatively low ranges. And that is reasonable because South China Sea is a relatively oligotrophic background. So 4.4 is, I think, just reasonable, and that means fecal pellets doesn't touch it. It's not the most important role when transferring the POC into the deep sea in our study areas. This is our first final result. As a second discussion, we want to talk about the changes of fecal pellets in the water column, about how what fecal pellets went through when it's sinking. So in this figure, I use a ratio of the parameters at 11.7 hydrometer over 5 hydrometers. So if this value equals 1, that means the parameter were equal at 5 hydrometer and the 11.7 hydrometers. And if the value was higher than 1, it means the deep water has more, and vice versa. So for example, let's first see the panel B. Panel B is about the FPN ratio of deep to shallow. You can see that most of the values are less than 1, especially in winter. Just only for two samples are obviously larger than 1. So that means the fecal pellet the number decreased with water depth, and that is, I think, is under the influence of the attenuation or the disruption when the fecal pellet is sinking in the water columns, it's gradually disrupted by like many mechanisms, and this is widely researched before. So, and the panel A is about the biovolume ratio of the over the deep to shallow, and this value we can see is the average value is most of them are large than 1, and that means for each individual pellets, the biovolumes increase with water depth. And we speculate there are two explanations. For one explanation is that there must be a new source to supply the particles and it supplies it provides some large fecal pellet itself, or it might be under the influence of in situ repackaging. And last about panel C. Panel C is about the FPC ratio. As we know, the FPC is a combination of FP and flux and the biovolume. So it depends on these two results. So for example, as we can see in the winter season, the number of fecal pellet decreased a lot. So this FPC ratio also decreased. So I mean, this is a FPC is a final result of these two factors. So our group made a very brief conceptual figure about how the fecal pellets going through when they were sinking in the water columns. First, we know that zooplankton excreted fecal pellets in the epitaxial zone in the shallow water, right? And during sinking process, it was fragmented through many mechanisms and to the deep sea. But in the maybe around 1,000 meter water depth in our study areas, there must be some supplementaries to provide the larger pellets. For as I said, there might be some deep dwelling zooplanktons and to directly excreted fecal pellets, or it might be supplied by the lateral direction, because our station locates very close to the Ganquan submarine platform and the China continental shelf. So the lateral direction might also be a great influence. So the last part I would like to we talked about the time series patterns in the fecal pellet carbon export, and we try to figure out what factors regulated the dark patterns. So I think the most convincing result is that in our study areas, the East Asian monsoon system has a very strong controlling factors on to control the seasonal variations in POC and FPC flux, and that was controlled by regulating the marine primary productions. As we can see in the peak of winter monsoon period, the sea surface water was cooled down, and the mixed layer depth was deepened. So that means the water was well mixed, and the nutrients can be reached to the surface water, and the primary production was higher in the surface ocean. So we get the result that in such winter monsoon period, the POC and FPC flux were high, and I think this is the most important regulate factors in our study areas. For other factors, we also make some speculations. For example, as I mentioned many times above, that once entering the winter monsoon period, the fecal pellets was was increased very sharp, very sharp. We don't know why. So we made a speculation that uh, can it be under the influence of migration of overwintering zooplankton? We don't have direct evidence. And also in the uh, late July to August, I have made some uh, other analysis, but I didn't show in this presentation slide. So there was a very huge mesoscale eddies coming through our study location. So I don't know, this might be influenced by mesoscale eddies. And also, as I said in the last slide, the lateral supply and advection from the continental coast and the continental shelf might give supplementary to the particles. But these two factors are we don't have solid evidence now, so it is our we want to figure it out in our next studies. So okay, I think here's all my presentation, and actually in recent years our team has reported several works about fecal pellets in different areas in the South China Sea. So mine is recently published in the western part, and my colleagues has also published in the southern and northern part of the South China Sea. And interestingly, the results show some kinds of differences, and I won't describe their work, and just show one of the results in the western part of my work. This is our field work. I have been on the ship for 3 years. And if you want to contact us, this is my email address, and this is my supervisor's address, who is also the corresponding author of these 3 papers. So yeah, I think here's all my talk, and I'm very sorry that maybe I didn't prepare this presentation so well. So if you have any questions or suggestions, I'm very pleasant to prepare. So, thanks for your attention.
Great, thank you so much, John Chan. That was really interesting and a lot of information to unpack there. So we have time for maybe one question. Does anyone have a question that they're looking to ask? Let's see, anyone in the chat? And feel free to post any questions in the chat if you think of one later. I just have a quick question. I know that I'm really interested in the sharp increase of the number of fecal pellets in May that you said. You said that didn't really know why. I was just wondering, do you have any I don't know, ideas of what it could be? It's really quite interesting.
Yeah, this so you can see this you mean this to sharp increase, right? Yeah. So for the first issue is that this is the last sample of this year's samples, right? So we don't know what is going to be like for the next sample, because this is our 1 year sample.
Right.
But however, if the next sample is also low, it means this is just an exception. I don't know, maybe I would like to explain it as a episodic lateral supply. I don't know, maybe maybe by now I don't have any clues because I have looked into the pictures I took. I have looked into pictures I took. Like this kind of pictures. These are my raw data. It doesn't show any kinds of difference. I don't know. It just there are more fecal pellets, and the shapes, the volume of pellets, they are just similar with others. They are just more. I don't know why they are more. So and I don't know. Might it can be the under the influence of eddies. I don't have clues now. Sorry. Maybe first I would like to get the next year sample and see what the next sample be like. It's also more or it's less. And if it's normal, then maybe I would check some like hydrologic parameters. I don't know, maybe rainfall.
Yeah. Or even if there's gelatinous blooms or something, that would be quite interesting. But yeah, no need to know, just quite interesting. So it'll be interesting to see that next set of data come back.
Mm-hmm.
All right. Well, thank you so much.
Okay, thank you so
for
much.
your time.
Yeah,
Great.
from my